I'm Chelsea Castrodale, representing the American Oil Chemist Society Young Professional Common Interest Group. I'm a food scientist with Coalescence Quality Chocolate, and I have a room full of young food scientists with me here, excited to learn about ice cream. I also have Brianne Crofts here with me, who works at CQC. She also starts as a master's student under Dr. Hartel next week, and she's going to be setting up our Babcock ice cream cam live for you. So thanks again, everyone, for attending our webinar. I want to thank the AOCS staff for coordinating all of the technology for Dr. Hartel and I. And of course, we want to thank Dr. Hartel for speaking today. Just an announcement, we'll be doing questions at the end of the webinar, and you can use your chat function that's in the GoToMeeting. You can use that to enter your questions at the end of the seminar, and we'll answer those live at the end. So this webinar was organized by the AOCS YP-CIG. The CIG aims to help connect young professionals within AOCS and help them navigate and get more involved in the society. We have events at the annual meeting, and we also have other resources such as this webinar outside of the meeting to engage our members. So if you'd like more information on the YP-CIG, you can check out the AOCS website and learn how to get more involved. So now I'd like to introduce Dr. Rich Hartel. Dr. Hartel has been a faculty member at UW-Madison for 32 years, conducting research on various phase transitions and teaching numerous courses on food processing and engineering. He has considerable experience in physiochemical aspects of fats and oils, including mechanisms behind partial coalescence. Dr. Hartel is also a past editor-in-chief of JAOCS. So with that, we'll let Rich tell us about fats in ice cream.
Great, thanks, Chelsea. Thank you all for showing up. Hopefully the next 45 minutes to an hour or so is an enjoyable time period to learn something about ice cream. And here's what the plan is. Talk a little bit about ice cream structure, so kind of set the stage for the main part, which is about fat destabilization or partial coalescence. We sometimes use those terms interchangeably. And then I've got a few things at the end on how fat influences quality aspects. And one of the interesting parts of this webinar, I think, will be following ice cream melting. If you were all in my lab or my building, I would have several types of ice cream up on the table in front, and I would have started them melting, and we would watch them periodically throughout. We're trying to do that through the two webcams, the one at AOCS and the one at Coalescence. And hopefully it works well. If you have some ice cream yourself, enjoy it, but it would be interesting to put a scoop aside just to see what happens to your ice cream, because we're going to go through how all ice creams are different, and yours may act very different from the ones on the ice cream webcams. So we'll start with maybe a popular idea about how bad Walmart ice cream sandwiches are because they don't melt. Maybe you've seen this set of YouTube videos where people leave their ice cream sandwiches out for hours in hot temperatures, and they just sit there. And they complain that the ice cream's not melting. It's probably because contains all kinds of junk and stuff like that. It's not really ice cream, blah, blah, blah. Walmart says, but it's a slower melt rate because we make it with more cream. Well, the bottom line is that ice cream has melted, but it has not collapsed. So that concept of melting and collapse, or the different concepts, is going to be an important one as we go through today. And so I'll come back to this idea at the end when we talk about the ice creams. From a structural standpoint, I think ice cream is probably one of the most complex systems that we put in our mouth. So certainly a multi-phase system with ice crystals as a dispersion, air cells, it's a foam, it's an emulsion because it's got fat, but ice cream mix has single fat globules. Hardened ice cream typically has a fat globule network that's made of clusters of fat globules. And this is certainly one of the things that varies from commercial ice creams. It's also got a serum phase in which all of the dissolved sugars, minerals, proteins, and probably hydrocolloids that are added to to give certain characteristics to the ice cream. And it's important to note that even at very, very low temperatures, there's some serum phase, there's still some water that's unfrozen. That serum becomes a glass somewhere about minus 40 degrees C. And if you cool below that, there's no more ice formation. So there's always some level of unfrozen water in ice cream, which of course plays a role in its characteristics. Let me pose another question, right? So years ago, you could go to the grocery store and all you could buy is Edie's Grand Ice Cream. Some people would find this as dryers, so depending on which side of the Mississippi you're on. But nowadays, you go to the grocery store and a big chunk of the cabinet space that's given to Edie's is slow churned. So the company that makes these Edie's has decided that they could manipulate the process to reduce the fat and maintain equivalent creaminess. That's the marketing argument that they have. So let me talk about how each one of these is made, because it's actually a processing development that allowed this to come about. So here's some answers. Slow churned has half the fat but tastes just as creamy. That's what they'll tell you. Slow churned costs more. That's absolutely true. They have different formulations to some extent, but they do have different manufacturing process. So the answer to this, of course, is all of the above. And here's the process. So the top one is Edie's regular ice cream, and this is the process that we use for the majority of ice cream that you buy. It's the mix comes in at this end of the scrape surface freezer with refrigerant taking heat out. The temperature of that goes down to -5 or -6 at the draw of this freezer, where about half of the water has now been frozen into ice. You might consider this to be soft serve type ice cream. It still flows and will settle, but it can be packaged and not leave air pockets in the package. This containers of ice cream then go into a hardening room where cold air is blown across it to bring the temperature quickly down to somewhere around -18 degrees C, at which point we say the ice cream is hardened, and then it goes into the storage and distribution system. And when we measured the average ice crystal size of Edie's full fat from the grocery store, it was about 54 microns. The slow churned, on the other hand, uses the same scrape surface freezer, but instead of going into a hardening room, it goes into an extruder, a cold temperature extruder that brings the temperature down from -5 to -6 down to -10 to -12. It can either be a twin screw or a single screw extruder, but the screw rotates at a very low speed, and that's where the slow churned comes from, is that those screws are moving at a relatively low RPM. And because of that, this process, the ice cream that comes out of here maintains smaller ice crystals, maintains smaller air bubbles, which change during the hardening process in normal conditions. And so when we end up with the product in storage and distribution, again, this is the mean size of ice crystals that we measured from the slow churn. So through mostly a process change, there are a few formulation changes as well, but mostly a process change, we're able to go from 54 to 34 microns. That smaller ice crystal tends to give a creamier taste. And so again, the marketing pitch here is that we can take out half the fat and still have as creamy an ice cream because the ice crystals provide some of that creaminess. Anyway, that's how ice cream is made. Also gives you some perspective about the slow churn product. So let's move to fat in ice cream. Again, in the ice cream mix, the individual fat globules are in the order of 1 micron or so in size. It comes through a homogenizer, and as it comes through the homogenizer, the new interface is covered with proteins. So you can see over here on the far left, the protein matrix caseins and whey proteins are very good at stabilizing the interface of this fat globule. But upon cooling and aging, the affinity of these surfactants, the small molecular weight surfactants, for that interface exceeds that of the proteins, and the proteins are replaced at that interface by these small molecular weight surfactants. Typically mono and diglycerides and polysorbate 80 is what we use. As it cools, of course, the internal globule crystallizes, and so what we end up with in ice cream is a series of milk fat globules that are partially crystalline And the interface is mostly devoid of proteins. And I thank my colleague and co-author, Doug Goff from the University of Guelph, for sharing his pictures like this, because they're pretty nice to see the fat crystals inside the fat globules and the surface mostly devoid of the protein, the casein micelles or submicelles. So this is what is in ice cream mix, these fat globules, again about a micron in size, partially crystalline in nature, that then go through the scrape surface freezer to make ice cream. So again, we have emulsifier added to ice cream, and usually when we add emulsifiers to foods, we're trying to emulsify or stabilize. Here, we're actually trying to destabilize the emulsion. The proteins are great at stabilizing. We add these small molecular weight proteins, and they destabilize that interface, take it from being something with a fairly high viscosity or rigidity or whatever term you want to use for that interface, and turn it into something that's now coated with these emulsifiers that have much lower interfacial tension and much less surface rheology. So that when we go through the freezing process under shear, these droplets are sheared and come together to form these partially coalesced droplets, right? So here's an example, again, from Doug Goff that shows two fat globules sharing the liquid phase, but unable to completely coalesce into a single spherical droplet because of the nature of the crystalline network inside this droplet. So we call that partial coalescence. And you can see that here, if we just had liquid droplets, when they come together, the liquid droplets would fully coalesce and we would just get larger droplets. When we have crystals inside, the crystalline network inside here often can prevent complete coalescence, and so we initiate coalescence, but it can't go to completion. So in other words, coalescence is arrested, and we end up with clusters of fat globules, semi-crystalline fat globules that are much larger in size than any of the individual fat globules. The old theory of how partial coalescence took place was based on some observations by Wallster, where he saw these, we call these lances, or really long fat crystals sticking out of a fat globule. And he never actually saw these lances penetrating another fat globule. He just started drawing pictures that look like this, where the crystals sticking through will penetrate the next droplet, then they'll start to share liquid, and we'll see those kinds of droplets. So for a long time, this was the theory behind which partial coalescence was based. You can still see publications that make this claim. And through a lot of work that we've done using micromanipulator system like this, we know that we can get partial coalescence without having those lances. But let me talk a little bit about this system. So this is a microscope. It's got micromanipulators. You can see them here. These hold capillary tubes that are in the shape of hockey sticks, if that means anything to anybody. And then these hockey sticks, which are pretty much flat level, go into an emulsion, a petri dish with an emulsion in there, and Abby now will collect a fat globule on one capillary and a fat globule on the other, bring them in proximity and coax them to do their thing. And so here's an example of two fat globules coming together, gently being coaxed to completely interact, and these are two liquid droplets, so they completely coalesce. But this is how we study partial coalescence with our microscope system. And so again, we can manipulate the conditions inside, and you can see in these two droplets, there's a little bit of crystalline structure. It's hard to see completely. You'll see some better in the next slide. But when we bring these two together, because of the crystalline network inside here, the interface will start to relax towards complete coalescence, but the crystal network, again, will prevent the completion of coalescence, so we've arrested coalescence. And as you can see, there are no obvious crystals sticking out of that interface. Even at much higher magnification, you can't see crystals sticking out of the interface. So we would argue, based on a lot of results that look like this, that the lances are not needed to cause partially coalesced fat globules. Here's a couple different things. This is a 30% wax inside a liquid fat, and you can see at 30% fat they partially coalesce, but the crystal structure again prevents it complete. Here's 40% solids inside there, and not surprisingly they come together, but because the crystal network is more rigid at that solid fat content, they can't coalesce nearly as much, and so the nature of the partially coalesced droplets depends on what the solid fat content is. And in fact, here's kind of an array of that. If you have 15% solid fat, and this is for this particular system, so each system is different based on the rigidity of the fat crystal network. And so the range of solid fat content where you get arrested coalescence varies for each fat, based again on the crystal network that's formed. So at 15%, there's not enough of a crystal network in this system, complete coalescence. But if you've got 50%, these two droplets just bounce off of each other. We've done some trials here where we would push one droplet up against the other one, and this one will, the other one will just completely separate from the capillary tube that would be over here because they're not going anywhere. They're acting like solid spheres. And again, depending on the nature of the solid fat content, we get different shapes of materials. We've also done this now with multiplets, not just two, but we can make 3, 4, 5, 6 different fat globules come together. Of course, the physics behind that is a little bit more complicated than this one, but it's pretty clear that we can make clusters of fat globules from systems like this. But we've been doing some work recently where we can make lances come out of a fat globule. And in this case, it's a pretty high level of STS and with high solid fat contents inside this droplet, we actually get crystals to come through the interface, which by itself is a whole interesting phenomenon. And then this droplet is really low in oil and solid fat content. So when you bring them together, you can see this lance starting to penetrate, and there is exactly what Walster was saying is true. And so under certain circumstances, those crystal lances certainly can promote partial coalescence. However, it's not always that simple. So here's another setup where, again, we have lances sticking out of this one, a low solid fat here, but these systems are covered with methyl cellulose. So this is 1.5% methyl cellulose in the solution phase, and we add that to increase viscosity so we can catch these droplets. Otherwise, they're hard to grab onto the capillary tubes. But the methyl cellulose has surface active properties and forms a nice rigid surface structure. So when these lances approach it, they just deform that interface and do nothing more. So under these circumstances, a lance does not penetrate into that fat globule and just sort of bounces off. But if you notice, this droplet has a little bit of solid fat content, but it's a heterogeneous fat. So it's crystalline over here and liquid over here. So now what we're going to do is take this droplet and put it over there, and then turn this one around and put it over there, and watch what happens when you penetrate the crystalline region of that droplet with the lances. And actually those crystals now at the interface disrupt the methyl cellulose enough that now we do get partial coalescence. We get sharing of liquid. Watch that liquid neck draw up. It's actually drawn that droplet off of the capillary tube, so there's enough force pulling it to pull it off, and then we get a partially coalesced network of a highly crystalline fat globule and a mostly liquid fat globule, but we needed the crystals at the interface to allow those crystal lances to penetrate. So some interesting things that take place there. How important that is in ice cream, still to be seen. This is something that we do on the microscope stage. So back to ice cream. Here's again some cryo-SEM images from Doug Goff, and you can see the fat globules surrounding the air bubbles, and we think that fat globules are preferentially adsorbed at that air interface, but not completely. They still form some in the serum phase, or the lamella between the two droplets, which is where ice would be, and all the serum phase as well. This particular ice cream, it's always confused me a little bit because you don't see a whole lot of clusters in this particular ice cream. And I think this is just one example of an ice cream that doesn't have a lot of clustered fat globules. And if I had to bet, I think I could predict how this would melt. So how do we get partial coalescence in ice cream? Well, it all happens in this scrape surface freezer. You think of it as ice formation, but also we have aeration taking place here. So air is injected into this end of the freezer along with ice cream mix. We get freezing taking place as the ice builds up. The shear stresses inside here build up, and that causes the air bubbles to decrease in size. And also causes the shear stresses that form from the freezing operation also cause those fat globules to partially coalesce. And so depending on the system, we can have a wide range of fat destabilization. So we have 20 to 60% here, but I've seen from 2% to 75 to 80%. At 2%, it means that most of them are still droplets, and at 80%, most of them are now in clusters. So in the 90 seconds that ice cream is in this scrape surface freezer, we have all of these things taking place. And it's pretty amazing sometimes, that what comes out of it is something that we like to eat. How do we measure partial coalescence? So basic light scattering method. And here's a typical trace of melted ice cream. If this was just the ice cream mix, this peak would be gone. We would only see the initial emulsion droplet and the casein micelles. But in ice cream that's got some of these coalesced clusters, we see a peak somewhere in the range between a few microns, this one shows 10, up to over 100 microns in size. So these are pretty large. I have a scale bar here, but this length is 60 to 70 microns in length. So fills in this peak. And so this would be an ice cream that has a lot of partially coalesced fat globules with still a bunch of small ones. You can see them in the background here as initial fat globules that have not gone through the partial coalescence. So to ice cream melting, and I can't see the webcams, but hopefully you can, and hopefully they've started to melt. But I'm going to show how two ice creams, and this is one of the ice creams that's on the Clausen plate. This ice cream, I think it's the same one that they have as well, but I'm going to start these. And this is how we measure ice cream meltdown, right? So it's a slab of ice cream. We try and keep size constant. Of course, if the overrun changes, then the volume changes. So that's another important factor. But we have a certain weight on here, and then we're going to put this, just hold these at room temperature and measure what drips through the screen down on the scale below here as a function of time. And we typically go for 2 hours, although now we go for 6 sometimes. So here's two ice creams that you can buy in the store. And again, these are, I believe, what the Clausen webcam is showing. I know that brand 1 is one that they have because we delivered it there ourselves. And brand 2, I think, is another one that I think it's this one exactly. So it's pretty clear that they melt in different ways, right? So brand 1 just melted and dripped all the way through the screen, leaving just this remnant of traces of the foam. This one, it's like those Walmart ice cream sandwiches, right? So they've collapsed a little bit, but it's still standing there 2 hours later. The first student that did this let it dry, and she would walk around with this screen and a dried hockey puck of remnant foam of ice cream on the screen, just to demonstrate that principle. So not all ice creams are equal. They don't melt in the same way, and not surprisingly, and now I'm going to relate that to the fat globule clusters. But it's not that simple, right? So meltdown of ice cream depends on heat transfer. How much air is in there determines what the thermal diffusivity of that system is. So heat transfer is a huge factor in how fast things melt. The ice content, that also influences the thermal diffusivity, because as ice melts, the insulation effect of water is much better than ice. And so again, there's an insulation effect from both melting ice cream or melting ice and the air bubbles. The serum phase viscosity is important because the lamella between the air bubbles is thinning. It's becoming less viscous as the ice melts, but the viscosity of that serum phase is going to determine how quickly it drains through this foam network. Of course, gravity is what the driving force for that change is, although buoyancy of the air might also be a factor as well. But then the fat globule clusters, their size, the numbers, the number density, is important in determining whether that melted serum can drip through this network of air or not. So here's a really pictorial schematic of, here's our ice cream melting on the screen, and we're collecting the drip through here, monitoring that as a function of time. But we can also look at the particle size distribution of what's down here and the particle size distribution of what's left up there, and compare it to the particle size distribution of the initial melted ice cream that we just put on the screen. And we always see what comes through as small fat globules, no clusters. What stays up on the top is where the big clusters are. So we start calling this the remnant foam, and clearly the remnant foam is where the large clusters of fat globules are retained, and that's in fact because of the structuring effect that we're going to see here. So here's a cartoon way of thinking about it, right? So here's an ice cream brand 1, you would say, that has a limited amount of partial coalescence. Most of these fat globules are individual in nature, and as the ice melts, the serum phase becomes more dilute and it starts to drain between these, and there's really nothing to stop it draining. And so those individual fat globules, which are 1 micron, which is smaller than the lamellar size, also drip through, and eventually you end up with just this remnant foam. Interestingly, if you put a slice of melted ice cream on a slide, this is what that structure looks like. So it's already melted, and because of the, it's not completely flat, you can see these fat globule structures in indeterminate in nature, but you can see them flowing easily between the fat globules, and that's what I mean about this serum phase draining between those droplet air bubbles, and so ultimately it just comes right through the screen. You can again see that with particle size distributions. So this is one that has only a little bit of partial coalescence or fat destabilization, and so there are a few and relatively small fat globule clusters left on the top. And what comes through is primarily the small individual fat globules, and you can see that in the light scattering pattern that the whole melt, which is the green curve here, there's almost nothing in terms of partial coalescence, so that's 5% fat destabilization. But what is there pretty much remains on the top, and the drip through is pretty much the same as, you know, there's very little in terms of what's there. So again, there's no structures to hold up that ice cream, and as it melts, it drips right through the screen. You can also see that in drip through curves. So this is the percentage of weight that's dripped through, but the height is the interesting one here, that it's collapsing, and then boom, it's all the way through the screen. There's nothing left on the screen. So again, this is one that has fast drip through, hardly any partial coalescence, no stand-up properties. These are new slides or new videos for us. I got these last week from the student that's working on it, and this is a melting video of ice cream starting at room temperature. And you can see, so this is one with low level of partial coalescence, so there's a lot of individual fat globules back here, and there are a couple clusters of structures here. So this one actually has a bit of structure, but now we're going to watch how these melt up to 40 degrees, and that's going to cause the milk fat to melt and allow the partially coalesced fat globules to fully coalesce. And that's kind of an interesting phenomenon to see. So first you can see the individual globules are flowing quite readily, but as you melt, the fat's melting and we're getting full coalescence. We also get some disproportionation of the air bubbles, which is kind of an interesting thing to watch. You can see that there's a lot of fat globules interacting at the interface. This one in particular has a lot of them there, but there are quite a few that are still just out in the serum phase. So this is in part what's happening when you put ice cream in your mouth. And I'll come back to that concept in just a bit. So now let's go to the other extreme where we've got a ton of these fat globule clusters, so a highly partially coalesced system. As the ice melts, the serum thins, and the system starts to collapse, but these large fat globule clusters are sufficient to form a structure in combination with the air bubbles that prevents drainage, complete drainage of that fully melted fat or fully melted serum system. And so it collapses a little bit, but then it just stays there, right? And so another way to think about it is that this jammed system of fat globule clusters and air bubbles has a yield stress higher than that of gravity once it reaches this point, so it can't flow anymore. And here's what happens again, melted ice cream on the screen. This is just at 20, so you can see that nothing's moving. And that's exactly what's happening in between these air bubbles. There's no drainage because there's no place for it to go. These particles, like a gel of yogurt, for example, that liquid can't drain through that matrix because there's just too much structure. And that's what's causing this particular ice cream to have that stand-up properties. And so you can see it here again, lots of fat globule clusters left on the top. What comes through is pretty much clean. You can see the light scattering. So the whole melt is this one in green. There's a ton of these partially coalesced clusters between 10 and say 80 microns or so. The drip through system has almost none of those clusters, and then the stuff on the top has almost all of those clusters and almost none of the individual fat globules. So again, it's easy to see these structures retaining the liquid melt serum as that ice cream melts. So again, it's melting because the ice is melted, but it's not collapsing because there's a structure that prevents drainage. You can see that in this one again, the height's coming down and eventually it plateaus. This was about 2/3 of its original height. So there's a bit of collapse and you need to have some collapse to get a jamming network set up. But so this sample, which would be brand 2 in our test, and hopefully it was one in the webcam test, is a product that has high partial coalescence, and so it just sits there and doesn't collapse. Here's what it looks like on the melting slide now. And so you can see a bunch of clusters of fat globules. There's a little bit of drainage in this one, but again, we're melting now. These fat globules are fully coalescing. There again is some disproportionation of the air cells. But again, you can see the same sort of thing happening as you put this in your mouth. And these two ice creams had exactly the same fat content, but the distribution of the fat was, one was in individual fat globules, and this one was in larger fat globule clusters, which will probably also have an effect on sensory, as we'll see later. How do we control partial coalescence? Well, the primary lever that we use is emulsifiers, and monoglycerides do some, but polysorbate 80 is really the key. So here's some pictures. Here's no added emulsifier. Here's added mono and diglycerides, but no polysorbate 80. And here's what happens when you add a standard ice cream mix of polysorbate 80 and mono and diglycerides. So it's usually 80% monoglycerides, mono and diglycerides, and 20% polysorbate 80. And the polysorbate 80 level is still on the order of 0.03% on a total basis. But you can see as you've increased the emulsifier level, well, the monos and dies have a little bit of an effect, but if you really want to generate partial coalescence, you add a little bit of polysorbate 80, and that is pretty good at the interface to promote partial coalescence. However, as I said before, the shear stress in the freezer is what's driving this partial coalescence. So anything that influences that shear stress is also going to influence the percentage of partial coalescence. And these are a few of the levers that we have to manipulate that. So going back to those samples, you should have seen a difference in meltdown. And we can think a little bit about what was different. And obviously, I would argue that fat content and the extent of partial coalescence, but overrun, the type of stabilizer that thickens the viscosity, and then the emulsifier that helps promote partial coalescence. All of these will have a factor to influence the meltdown rates of ice cream. And so the next slide here, you see a commercial evaluation. So this was us going to the grocery store, buying a whole bunch of ice creams, measuring drip through rate, measuring fat destabilization. And yes, there's a general trend that drip through goes down as you go to higher fat destabilization. There's a lot of other factors that influence that it's hard to get from commercial products. We've done a bunch of work, Maya Warren in particular, trying to control things in more details. So let me finish off with a couple slides on sensory. And so one of the things that came out of the commercial study was that the ice cream with the largest ice crystals was not rated the coarsest, but it was also the one with the highest extent of fat destabilization. So the next student that I had figured out how to make ice creams with the same ice crystal size, but different levels of fat destabilization within a range. I mean, we couldn't make 70 micron ice crystals with 60% fat destabilization with the conditions that we were able to use, but we were able to go from 0 fat destabilization to 15 or so. And look at the sensory score in iciness decrease substantially, significantly, as a function of how much partial coalescence is. So the particular ice cream that I'm talking about, if I can go back, I think it's this one here, but don't hold me to that, and you don't have the codes anyway. But it had 60% partial coalescence, and if you extrapolate this in some way, you know, its iciness score would be really low, which was exactly what we found in that sensory analysis of the commercial products. And so we talk about, well, what's the threshold size for ice crystal detection in ice cream? It's not so simple. So the answer is it depends. And one of the things it depends on is the iciness or the fat destabilization rate. The second parameter it depends on, and I'm not going to show the data, is the viscosity of the continuous phase. So the stabilizers added to ice cream also significantly influence iciness score, sensory iciness, for products made with the same ice crystal size. So interesting correlation between fat globules and fat destabilization and sensory of ice crystals. And then let me return to the meltdown. And one of the interesting things out of that study was, you know, we had drip through rates from the screen, and we had sensory melt rates in the mouth. And we sort of asked people to do the same thing in their mouth. Well, not really. We couldn't have them sit there and let it drip through their tongue. So it's not quite the same thing. Plus, there's a warmer environment, so the fat's melting, like we saw before. We've added saliva to the equation now, so it's thinning things out by itself. And then shear forces. We asked people to just put it on their tongue and let it sit there and see how it melted, and compare how the different ice creams that we gave them melted. And interestingly, there's no connection, no correlation whatsoever. So I think future work will probably start looking at meltdown tests that might better correlate to what happens in the mouth. So let me wrap up here before I hit you with one final question. And so I've tried to convince you that the fat content is a pretty important parameter that influences meltdown and collapse of ice cream. And I just want to emphasize again that melting and collapse are two different things. So when people say, oh, that ice cream didn't melt, it's been sitting out at high temperatures for 5 hours, you should correct them that no, the ice melted, it just didn't collapse, right? And so that's what the Walgreens ice cream sandwich is. And then there's these commercial Japanese popsicles. I don't know if you've seen this. There's a lot of things going on with that where the same phenomenon is taking place, where they say it holds its shape up to about 40 degrees C, but it's melted. It just isn't collapsing. There's some interesting stuff in that as well. So finally, ice cream, a pretty complex food, and the fat is a pretty important part of it. So working on fat and ice cream has been a pretty enjoyable segment of my research. And then I'll just finish with this one, because you don't have a good talk unless you can tie it to the OJ trial. Maybe some of you are old enough to know the OJ trial, but there was some ice cream left on the banister. It was Ben & Jerry's left on the banister, and the lawyers were trying to make an argument about meltdown of ice cream, how fast it melts, and whether OJ could have been there or not. And they argued that it wasn't totally melted by a certain time, and he couldn't have been there because he was already on the way to the airport. So what would you tell the prosecutors? Yeah, hopefully you would say, well, meltdown is different from collapse, so go away, it doesn't prove anything. And with that, Chelsea, I'm open to questions.
Great, thank you so much, Rich. That was really interesting and a delicious treat of Babcock ice cream here on our end. Thanks again for sending that over. And also we did have the Babcock ice cream and we had the Dean's vanilla. So I believe we did have the ones from your experiment.
Okay.
And the AOCS cam shows the ice cream sandwich you referenced.
Right. Right.
Yeah.
And so it's interesting that the one on the left here, the non-Babcock one, didn't show that much difference from the Babcock ice cream. So very different from when we do the test. So it's possible that you know, that doing it on a plate is different than doing it on a screen. It's possible that, so the original ice cream we did that experiment with changed over the years. So whether they changed their formulation or it's in their process, I don't know. So things are not always as consistent, except for Babcock ice cream. It's always melted.
So we can open it up for questions. You can use the chat function on the GoToMeeting to enter your questions, and I'll read them off as they come in. So we'll wait for those to filter in. But I have one as we're waiting. So, I mean, I experienced this when I was trying to prevent this when I bought these ice creams the other day. Well, not the one you delivered, when I bought the Dean's. So once your ice cream has melted slightly, maybe as you're driving home from the grocery store, is it possible to achieve the same physical properties again when refrozen, or is it just, it's gone?
It's gone. So once the ice melts, you know, we spend a lot of time controlling ice crystallization in that scrape surface freezer. And as soon as it melts, when it refreezes, it goes back under uncontrolled conditions. And, you know, another example of that is, yeah, I do this too, right? I take the ice cream, and even a pint, because a pint's not a single serving, is it? And I take it out, and it's too hard to scoop, so I either let it sit on the counter for a few minutes or I put it in the microwave. And then you put that back in the freezer after you've scooped out your dose, and then you come back the next day and you see these ice crystals growing out of the surface. Those are, let's see, G for ground, so those are stalagmites of ice growing out of the surface of the ice cream. And it's because you've melted the ice and now it's going to refreeze in a way that best suits it thermodynamically. And that's not in the form that the scrape surface freezer does. And in fact, let me throw a quote out because it fits, and I like saying this story, right? So heard an old time ice cream maker, actually equipment maker, talk about ice cream once at a conference. And he said, ice cream at equilibrium is dead and should be buried. And what he meant was, you know, we have this fine dispersion of ice crystals, and at equilibrium we get one large ice crystal with the minimum surface area, and we have all these small air bubbles, and at equilibrium we'd have one large air bubble to minimize the surface energy. So that's the nature of things. Thermodynamics kind of works against us, and that's what's happening when you melt your ice cream and try and freeze it.
Great, thank you. We have a lot of questions rolling in now. Can you explain what sensory iciness is for those of us who are not familiar?
Well, we've probably all felt it. Cars, you probably have too, right? You put the ice cream in your mouth, and the ice crystals are so big that you can feel them on your mouth. And so it's a coarse perception. It's not that's different from the sandy perception, which is what happens when lactose crystallizes. But because ice crystals melt so fast, the threshold detection size for ice crystals, we say, is about 50 microns. But for lactose crystals, it's about 15 microns because of the difference in dissolution rates. So it's hard to say anything more other than you can feel the ice crystals in your mouth. You want a really good experience for that. Make ice cream at home in a brine freezer. Eat some right away because it's delicious, right? But put some in a pint and put that in your freezer overnight, and then taste that the next day. And that's the definition of coarse ice cream.
Okay. Does higher fat destabilization and the lower drip through rate mean that the ice cream will be better or less icy after sitting out and refreezing?
So I don't think so. I think this sort of goes back to your question, Chelsea, and thanks for that, Jill. I don't think so. I think the ice will still tend to go to its thermodynamic shape, which, you know, it's going to fill the space that the water is in, and it's just going to make big ice crystals. Will those clusters get in the way? Maybe slightly, but it doesn't form a strong enough network like a yogurt would to prevent the ice crystals from getting bigger. So I don't think so. I've never done it.
Okay, we have one here from Dr. Marangoni. What characteristics should we look for in a fat for optimal induction of partial coalescence? Why is milk fat so effective versus others?
That's a good question, Alex. And I talked about solid fat content, and that's an important part. And I think milk fat is kind of right in that, you know, that zone where you get the right conditions. I think coconut fat is probably there as well, but you wouldn't want to put cocoa butter in ice cream because the solid fat content would be so hard that, you know, as in our micromanipulator experiments, you would never, shear forces wouldn't be high enough to cause them to start to coalesce. And if the solid fat content is too low, then they're just going to completely coalesce. And actually, if you look at some of the non-dairy ice creams that have used some other fat, some of them actually use a liquid fat because, you know, they're afraid of the saturated fats, and those have very large fat, can have very large fat globules that have coalesced at some point completely. And I don't know how much of an effect does that have on texture. Boy, we don't really know. You know, those non-dairy frozen dessert products aren't new enough that we've got a lot of understanding behind it. But beyond solid fat content, you know, the surface. So I'm inclined to say surface rheology because that's what everything is pointing towards. But certainly the surface structure influences that as well, and then the shear stresses in the freezer influence the level of partial coalescence as well. It's pretty complex, as I was trying to say.
Yeah, we actually just had the question come up here in our room at Clausen about non-dairy ice cream, so thanks for touching on that as well. Okay. When measuring particle size of fat droplets in ice cream, can you measure it on the melted ice cream post-freezing rather than on the ice cream if you don't have a cold stage microscope?
We use a Malvern mastersizer to get those curves, and the straightforward thing to do is to take ice cream mix, dilute it to the right level, and then put drops of the diluted mix into the cuvette of the mastersizer to reach the right scattering sense there, and then you just run run the light scattering on it. For ice cream, we would melt the ice cream, dilute it again simply with distilled water. That by itself allows the air bubbles to come out, so we're not measuring the air bubbles. But the fat globules, even at room temperature, fat globules are not changing very much because there's still enough solid fat in milk fat to allow you to see it. So we just do it at room temperature, and we believe that what we're seeing is correct. Certainly for milk fat, again, as you go to other fats, I'm not convinced, and I think melting of the fats is pretty important to getting that. So we have a little bit of a side project going on non-dairy stuff, but not enough results yet to be able to say that. So we take pictures primarily to back up the light scattering measurements, right? So I was always taught that if you use an instrument to measure particle size, you better back it up with a microscope just to make sure that it's not an artifact. And that's why we do that.
So we just talked some about non-dairy. Now we have a question regarding using different sweeteners. So how are ice cream properties changed when using polyols rather than sugars?
Halo top is a pretty popular thing these days, and they use erythritol as the sweetener. And so there's two factors that go into that. In order to have ice cream that's semi-scoopable at freezer temperatures, you need to have the right freezing point. And so you've got to balance freezing point depression with your choice of sweeteners. So that's important, but the other factor is viscosity. And typically polyol, certainly erythritol, has a much, much lower viscosity than sucrose. It's a monosaccharide versus a disaccharide. You can manipulate that with hydrocolloids, but if you're going for a clean label, you've got to use something that fits under that category as well. So probably the most important things are freezing point, which you can manipulate in a variety of ways, and viscosity, which you can also manipulate in other ways. So I don't think the polyols play that much of a role in fat destabilization, maybe a little bit from the viscosity of the serum phase, but that's about it.
Okay, we have former AOCS President Neil Whitlock chiming in. If solid content is important, is crystal morphology also important?
I think absolutely so. And as I said, for the different fat systems that we've played with, the range of solid fat where we get that, the dumbbell shape, the middling of partial coalescence, changes. And, we've related that to the rheology of the bulk fat. It's hard to measure the rheology in, even 5 micron droplets, much less 1 micron droplet. But we think the correlation is there. So it's not just crystal morphology or polymorph, it's how the network forms. And so the rigidity of that crystal network, I think, is what controls that. And we have a model that we think predicts that, but actually it doesn't work for all of the fats that we've used. So there's something more still in that that controls the partial coalescence and where you get it and where you don't. So I wish I had a better answer to that, but that's still an open question.
I had a kind of more of a consumer-driven question. Do you think with for example, the Walmart product not melting, do you think that was developed as a desirable characteristic so it wouldn't like melt and get all over a small child, or is it just a fact of probably a lower cost and lower quality formula?
So in my opinion, that's giving manufacturers too much credit. I don't think most ice cream manufacturers consider that. And certainly they don't know in general how to control the levers that control it. My guess is that the bigger ice cream companies probably do. I know that some bigger companies have been looking at that for a long time, but because of what we see in the commercial products out there, they're all over the place. And, again, I know that the people here in Babcock have no clue, even though we show them this data all the time. They don't care. One of the other ice creams the one that had the largest ice crystals and the highest partial coalescence is also a local ice cream. I'll tell you later, Chelsea. And I guarantee you they have no clue either. So they just don't know what's controlling their product. And I'll quote the pilot plant manager here, says that I don't care as long as they keep coming back to buy it. That's his perspective.
Yeah. Are there any other questions for Dr. Hartel? Just not seeing any more coming in. Well, then I think with that we can wrap up. I'd like to thank you again, Rich, for your time and for preparing all this for us. And We want to thank the AOCS staff for organizing and getting us, getting all our tech in check. And thank you to all of you for joining our webinar today. And we look forward to having more AOCS YP-CIG webinars in the future. So go enjoy some ice cream and have a great day, everybody.
And good luck to the AOCS group cleaning that stuff up.
Oh, yeah. Thank you.
Take care, everybody.